Hello everyone. I present today our work on a novel constitutive descriptor of fibrous extracellular matrix mechanics and remodeling. I will also discuss the steps we are taking at the tissue mechanics and remodeling lab to further this work and the positive implications of this study. Many biological processes such as growth and development rely on cell-generated mechanical forces to remodel the local ECM. These cell-ECM interactions are commonly studied in vitro using networks of type 1 collagen as shown in figure 1 that shows two breast cancer cells in red and collagen in green. Adhered to the ECM, the cells contract and remodel the network to align and stretch the fiber around the shortest path joining them. While imaging and measuring the structural consequences of this remodeling has become feasible today thanks to innovations in optics and microscopy, accurately quantifying the mechanics of remodeling fibrous networks remains an unmet challenge. To demonstrate that fiber-based constitutive models represent network mechanics more accurately, we performed finite element simulations of contracting cells in ECM based on published data. We compared results obtained from modeling the ECM as a non-fibrous neo-Hookean material and a fibrous strain-hardening Wang-Shenoy material. Figure 2 shows that the displacement field produced in a Wang-Shenoy matrix is long-ranged and propagates to a distance of around 10 times the cell radius. Figure 3 shows that the displacement field becomes even longer-ranged if we account for cell elongation, here modeled by the factor alpha, and the contraction anisotropy, here modeled by the factor beta. We note that the effect of anisotropy in shape is more prominent. The Wang-Shenoy model represents the state-of-the-art in continuum modeling of fibrous networks. However, the model is completely phenomenological and its material parameters are obtained from fitting experimental data, and they do not rely on the structural changes that underlie the remodeling process. In this work, we propose a model that incorporates the microstructure of the collagen network. 3 of the 5 parameters of our model, the fiber density, the diameter, and the length, are structure-based and can be determined accurately via microscopic imaging, leaving just 2 parameters C1 and C2 to be determined from mechanical experiments. The mathematical formulation of the model is rooted in the nonlinear theory of elasticity. For a representative volume element shown in the schematic, we average the contribution of individual fibers to calculate the energy stored in the fiber network as a whole. We model individual fibers as nonlinear elastic springs described by an exponential strain energy function. On the right, figure 4 shows 2 photon microscopy images of a collagen network which are analyzed using CT fire to obtain fiber density, diameter, and length. Planar biaxial tensile tests were used to inform the parameters C1 and C2. The material parameters from our analysis on 2 and 4 mg per ml collagen gels are listed in the table at the bottom right. This work demonstrates the feasibility of combining structural and mechanical data into a model that is capable of describing the remodeling process. Next, we propose to combine the constitutive model we have developed with a microrheological method based on magnetic twisting to study the remodeling of ECM by invasive breast cancer spheroids. As shown in figure 5, ferromagnetic beads are embedded in the collagen matrix surrounding a cancer spheroid. Under the action of an externally applied magnetic field, these beads twist to generate local displacements that can then be measured using digital volume correlation and recreated using finite element simulations to inform the parameters C1 and C2 of our model. This study will enable us to establish a reliable microrheological approach of quantifying tissue mechanics in a spatially and temporally resolved manner. This knowledge can then be used to better understand the physical basis of cancer invasion and can be applied to study other processes such as development and morphogenesis.